Well, hello. Welcome back, or welcome to Physionic. Today's going to be a shorter, long-form video, mainly because we only have a single piece of data that we're going to be going over. But it's interesting, it's intriguing. We're going to be discussing how carbohydrates affect our mitochondrial function. And this comes from a branch off study that came from a main piece of data or study that we actually covered several months ago, and then I just recently released a short form of that study looking at how varying levels of carbohydrate consumption in human beings leads to some interesting metabolic effects, interesting effects on our metabolic rate, our metabolism. So if you're interested in that, certainly check that out as I release more and more content for that. I think I have about 4 videos, maybe 5 videos lined up for that series, going over a number of different studies to investigate how carbohydrates affect our metabolism. Now, I am covering this as kind of an offshoot of that series. I don't think I'm going to actually add it to the totality of the series, but it's an interesting little tidbit on the side, and it offers some clues on how carbohydrates affect our mitochondria. And of course, de facto, our mitochondria affect our metabolism. So that's what we're going to do today. If you're not familiar with who I am, for those of you that are new, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. I actually work in a mitochondrial laboratory at the moment, and although I have done I've done these measures that we're going to be going over. However, I don't routinely do them because I do other types of measurements or experiments other than the ones here, but I have done them in the past, so I'll be able to describe to you what the researchers are actually doing and what this tells us about our mitochondria specifically. Okay, with that though, let's go ahead and jump into this, and we'll get this through pretty quickly. All right, so as usual, the question that we are examining is, how do carbohydrates affect mitochondrial function, more specifically energy generation? Because if you've been following Physionic for a while, you know mitochondrial function is usually defined by a number of different things, but in this case we're talking about energy generation, or kind of mitochondrial associated metabolism. Now, this data comes from a paper out of the European Journal of Clinical Nutrition, and it's called A Brief Carbohydrate Diet, or a high carbohydrate diet, lowers the rate of adipose tissue mitochondrial respiration. All those words will become quite clear to you in short order. Now, to introduce this, before I actually get into how they did this study, I wanted to quick mention that apparently, according to these researchers, which I certainly believe them because this is just a quick history lesson, where some of the ideas for this experiment, this study, as well as the other study that I covered that was far more expansive, the idea came from a historical discovery in 1912 where diabetic individuals, type 1 diabetic individuals that have struggle or do not produce insulin for themselves because they're, for various reasons, but their pancreas just doesn't generate insulin anymore. When testing their metabolism, researchers found that those individuals had an increase in a metabolic rate. They had an increased metabolism from lower or non-existent insulin. So, and this was correctable, as in they could inject insulin, of course in a controlled situation, inject insulin, and these individuals' metabolic rate would then normalize or stabilize or decrease back to what was expected or what was calculated that their metabolism should have been based on, a variety of different factors. So a lot of that was based off of the mechanism of action was thought to be the effect that insulin has on mitochondria. And I already have videos on that specifically where I discuss the data actually from a similar research group as the one we're going to be going over today that did try to figure out how insulin affects our mitochondria and a little bit beyond that as well. So if you're interested in that, I will link that as well. But I'll be adding that series of videos as well to the overall series on carbohydrates and metabolism. So that's kind of the backstory of how they came to this idea of looking at mitochondrial respiration. Again, something that you may not know the term for, but I'll explain in just a little bit. All right, so to discuss the study design, we're just going to use this one diagram. So they had, this is, the reason why I said this was an offshoot study of the other study that we've covered is the fact that they just took samples that they were already going to be taking for the other study, for the main study, and they decided to kind of have an offshoot experiment and not add it to the other study, but actually just create a new study, lower impact study. So what they did is in this other examination, I don't want to keep saying the word study because it gets repetitive, but there aren't that many synonyms for study. Paper, investigation, things of that nature. Well, all right, so I came up with a few. So they recruited something like 27 overweight individuals, men and women, and they were split into 3 groups. And you may, if this sounds familiar, that's because you've already heard this from when I went over the other investigation. So one condition was a low carbohydrate condition, was 20% of their diet was carbohydrates. Then they had a moderate condition, which is 40% of their diet was carbohydrates, and a high carbohydrate condition, which was 60% of their diet. Now, for transparency, the high condition did have slightly more women in this condition, and obviously fewer men, than the other 2 conditions. So that could be a confounding variable. Do I think it is? No, but that's up to you. If you think that could play a factor, if you have some sort of rationale or some reasoning, some mechanism of action that would potentially lead to some doubt in terms of the results of this study as well as the other study. And then what they did is, as displayed here, what they did is they took fat biopsies. So they took fat biopsies, meaning that they took cells and then plated them onto a cell culture dish. And actually, in this situation, they may not have, now that I think about it. They may not have added it to a cell culture dish, but they took the biopsies and then they froze the biopsies so that they could do experiments on them. Or they did the experiments right away. And the reason why I'm kind of juggling it in my head, because there are a number of different ways of doing it, but I believe the way that I'm describing it now is correct. They took the fat biopsies, meaning that they take some tissue that's mostly fat, that's taken obviously from a fat site, and they took it before these individuals were on a diet and then afterwards, but we're going to be focused more so on the afterwards. So we're going to be focused on the comparison between the individuals that are consuming a low carbohydrate or lower carbohydrate diet, moderate versus high carbohydrate diet. And their calories were roughly the same. And then what they do is they permeabilize the cells, meaning that they essentially break open the cells. So all the cells, break open. And then from there, they're able to extract the mitochondria from these fat cells. And then they run mitochondrial measures on these mitochondria. So some bit of background information that you should know. Let me get rid of my face before we get into the single piece of data that we have is, and again, if you've been following Physionic, I've been covering a lot of mitochondrial work because I'm getting deeper and deeper into my dissertation. So I have to focus a good amount of my reading because I've in the comments, people seem to be surprised that I have 3 jobs. This is a job, reading research specifically for Physionic, then creating content and all that. And then I'm a professor, so I teach at another university, and then I also have to finish my PhD, so I've been working in the lab for that as well. So it's draining, so I can't always do dedicated content to physionics. So hopefully it being a little bit more mitochondrial heavy for every now and again is okay with you guys. But I digress. So let me give you a bit of background on the mitochondria. Here we've got our single mitochondrion, and if we were to zoom into this little section right here, we would find that there's an outer membrane of the mitochondrion, an inner membrane of the mitochondrion with an intermembrane space. Again, if you've been following Physionic, you know all about this already. But I'm not going to go into too much specific, but we do have to have a little bit because this is going to give us information on how to interpret the data that I'm going to be showing you. So normally what happens is you have these molecules that come from our food. So food is converted to a series of other molecules, which eventually get converted to NADH and FADH. And these two molecules will donate electrons. You don't really need to know what an electron is. Just know that they donate something, which is known as an electron, to these different proteins that are part of a complex of proteins, right? There's 1, 2, 3, and 4 are the main ones, and then the ATP synthase is often considered complex 5. So complex 1, 2, 3, 4, and 5. And then there's these other accessory proteins that are involved as well. So anyway, these molecules will donate electrons to these proteins, and these proteins will then push the electrons from one protein to the other protein, et cetera, until it gets to complex 4. Now in complex 4, it gets ejected and used to generate water. So it uses oxygen and it uses hydrogen and uses the electron to generate water. So this is where we get the idea of mitochondrial respiration. What they say in the title here, mitochondrial respiration, comes from this use of oxygen here. So obviously, if you have a lot of electrons moving through here, you need a lot of oxygen to be able to produce this metabolic water. So the faster, the more activity you have here, the more water that you're going to be producing, and really what we're interested in is the more oxygen you're going to be consuming, meaning that you have greater activity. Now, the ultimate end result of all this is that you generate ATP. So you generate cellular energy as a result. Now, I'm glossing over some details here, but you don't need to know too many more details for the moment. So this is how it works. Electrons get donated, they get pushed from complex to complex, then they get ejected at complex 4, and you get the consumption of oxygen to generate metabolic water. And then you get end product of cellular energy. Now there's a bypass system to this entire thing known through a drug known as FCCP. And FCCP will create a leak here that leaks these protons, these H's up here, these hydrogens, which are also known as protons in this scenario, and will allow protons to leak back into the mitochondrial matrix. Now, the reason why that's a problem is because this ATP generation requires protons to move from here to here, from the intermembrane space to the mitochondrial matrix. But you want it to go through the ATP synthase, not to go through a leak right here. I used the hose analogy last time that I talked about this. The hose, the end of the hose where the water is supposed to be coming through is this ATP synthase. But if you start poking holes in the hose, then you have less water going through the end of the hose, and you have more coming out of the sides of the punctured holes of the hose. That's FCCP. Now that's perfectly fine because that ends up being used as a tool by researchers as long as you control the amount that you use. Too much FCCP will end up killing the cells and end up killing the mitochondria. But we don't care in this situation because we've already killed the cells. The cells are gone. We're just looking at mitochondria. We've permeabilized the cells. We're just looking at mitochondria. So FCCP will allow this leak of protons. And as a result, this has to function a lot faster because these are pumping protons back out to the intermembrane space from the mitochondrial matrix. So this has to go super, super fast because there's this added leak as opposed to just going through this one controlled mechanism. And I'll explain this a little bit more with the next slide. Okay, so to look at the data here, I've got things labeled here. You can see my face again. I've got room. So you can see the data here. And this is mitochondrial respiration. Again, oxygen. How much oxygen? They're just measuring one thing, but it tells you a whole lot about this entire complex. And that is how much oxygen is being taken up by the mitochondria to enact this entire process, to allow the electron pushing, to allow the protons to be pumped back into the intermembrane space, and to allow ATP generation. So here we've got our basal scenario, and I realize that they don't have indications for what is what. So white is a low carbohydrate situation. So low carbohydrate, the fat, the mitochondria from the fat of low carbohydrate consumers, so 20% of their diet. This is the moderate, so the gray is the moderate, and then the black, as you can imagine by the process of elimination, is the high carbohydrate condition. And while this does look different from one another, right? If you were to compare this bar to this bar to this bar, you would see, or if you just look at it without even realizing they're bars, you just think they're different from one another. But it turns out that statistically they're not. So these are the same. And this is a basal condition, meaning that this is the GM. So let me explain that a little bit. So this is all part of a mitochondrial assay, and the GM is a glutamate plus malate. So those two molecules, glutamate and malate, are added to just the mitochondria, the isolated mitochondria. And then they measure the amount of mitochondrial respiration that occurs. So glutamate and malate actually end up allowing for the generation of more NADH, which is what I mentioned before, that interacts with these first complexes to donate their electrons. So GM, which is here in my diagram, allow for the greater generation of NADH, which then allows for more electrons to go through this system. However, when you supply more NADH substrate, it doesn't do anything. There's no difference. So basally, there's no difference, okay? Now, GMD in this scenario right here, we're adding ADP, which is right here. ATP, the cellular energy molecule, is generated from ADP, which is the ATP synthase takes an ADP molecule and a phosphate, slams them together, and ta-da, you got ATP. So the researchers are specifically they're still adding this glutamate and malate, but they're also adding ADP to the mitochondria. And they're seeing, is there an increase in mitochondrial respiration? And it turns out that, yes, there is. There is an increase specifically with the lower carbohydrate conditions compared to the higher carbohydrate conditions. Now, interestingly here, this did not reach significance. So the lowest level of carbohydrate did not reach significance, at least based off of how I'm reading this. However, here we're starting to see a picture form in that the moderate carbohydrate condition did see an increase in mitochondrial respiration specifically over the condition or the individuals, it's a mouthful to say, the mitochondria from the fat cells of the individuals that consume a high carbohydrate diet. So there is an increase in respiration compared to those individuals. I'm just going to call it the high carb group. High carb, moderate, low carb group. So there is a difference here, and that's just with the addition of ADP at this point. So here we're seeing a difference that's a step further from the basal state. The next thing that they wanted to look at was, okay, well, what if we stimulate not just at the ATP synthase right here, not only at complex 1 right here, but what if we also add succinate I believe, yes, it was succinate, which allows for the generation of more FADH. So not just NADH generation, but FADH generation, which means even more electrons are being donated through a different complex this time, complex 2 as opposed to complex 1. And what do they find? Well, they find that there is a significant increase in mitochondrial respiration in the low carb and moderate carb conditions, but not in the high carb condition. Hopefully you're seeing this trend now, and you can interpret this last one on your own, because it follows the exact same general principle. However, in this situation, they're using FCCP, so they're not stimulating through these different complexes. What they're doing is they're using this drug that I mentioned earlier. So the FCCP will create a leak, and this mitochondria doesn't. I don't want to say it doesn't like, because that's personifying the mitochondria. But generally, you do not want, or the cells generally don't want, or the mitochondria don't generally want a lot of protons in this inner mitochondrial section, the matrix. So FCCP creates this leak where you have constant protons finding themselves in the inner section of the mitochondria. So clearly, as a result, these complexes have to really ramp up to pump all those protons back out. So it gives you, it's a mild stressor that allows you to measure the maximal rate, the absolute maximum rate that these mitochondria can function. And what do we find? Well, we find that respiration again increases for the low carbohydrate and moderate carbohydrate, but does not seem to increase for the high carbohydrate group. So pretty interesting, pretty fascinating. The researchers did take certain liberties in their interpretation of this data, which I will be correcting, at least from my perspective. All right, so to conclude here, I told you that this was going to be relatively short, although I've been blabbing on for 22 minutes now. When I'm done editing, it'll be like 20 minutes. A high carbohydrate diet reduces mitochondrial respiration, which means that it reduces energy production or the consumption of oxygen, as I explained. Now, here is where things are a little bit different from how the researchers interpret this. They may have stretched things a little bit too much, I think, in my estimation. So a high carbohydrate diet reduces mitochondrial respiration. We saw that. So mitochondrial activity, energy production. And the researchers then end up implying that this might lead to greater fat deposition. So because you have less mitochondrial activity, then where are the nutrients and stuff going? Well, they may end up being deposited in the fat cells. But I don't feel that that's a fair conclusion, because right now we're just measuring mitochondria in isolation by themselves. And it does tell you some cool information, and it tells us some mechanistic information, but until we actually measure the fat deposition itself, I think it's a stretch to say that, oh yes, that's definitely happening. That's why I said it's a clue. It offers a clue. And we're going to be going over more on that particular topic. I've already have some content on how insulin affects mitochondria and fat deposition and things like that. So certainly check that out. But as we continue through this series, as I release more videos on the topic of carbohydrates and their effect on our metabolism, we may uncover a few more concrete pieces of information that we can actually take away from this. Okay, so hopefully that made sense to you. Hopefully, if it didn't, please feel free to ask questions. And with that, I will leave you, and I hope that you have a wonderful, wonderful day. Have a good one.